Hello, everybody. I'm not sure if everybody's here, but let's start anyway. So, my name is Sebastian Bludau. This is Timo Dickscheid. You've already seen him during the first session. And we would like to give you an overview about eBrain's tools and also atlases, which are included in those eBrain's tools, and some Python scripts in the second part of this hands-on workshop. So it's generally designed that we have two parts. We will start with a very basic overview about the concept of the atlas and about the atlas viewer and how to use it. And later on, we will skip to a Python part. Both sessions, both parts are planned to be to have hands-on sessions, hands-on parts. But let's see how everything works. In general, it would be good if you are all connected to the local Wi-Fi so that you can try the examples in the Explorer on your own. And there are also some printouts, which they are currently here in the last line, with some of the basic URLs you will need to find the tools we will show you. So, as I said, starting with an, everything fine? General overview about the concept. So it's a very dry slide, but this defines the concept, how we see and use our atlas and how the wording is used. This is quite important because, especially for the second part where we use the SIIBRA Python, you will use atlas, parcellation, and reference spaces as keywords to get the data you would like to work with. So in general, it's comparable to a normal map, to a normal atlas we know from geography. So we have atlases. For example, it can be a human atlas or a red mouse. We have different atlases included, comparable to different planets. Those different atlases include parcellation schemes. So you have different parcellations. For example, the BigBrain atlas. This is the one we will concentrate here on this workshop because it's coming from our working group. But of course, there are different parcellation schemes included. And this is comparable to countries, continents, time zones. It depends what you are looking for, what kind of parcellation you would like to use to apply to your atlas. Then we have different reference spaces. Those are very important because if you want to share data, if you want to compare your own data with the results from different labs, then you have to agree on a reference space so that you can you have to bring your data in a reference space. And once you are there, you can use different data modalities to compare them directly. And this is comparable to different coordinate systems or projected coordinate systems if you want to compare it with the normal atlas model. And then we have, like you have it in Google Maps, for example, if you browse to a city, you can also click onto a restaurant. You can see when it's open, how the prices are. So, and those are regional features. So we also have cell densities or receptor densities, functional data, all those data sets which belong to a specific region of a specific parcellation scheme in a reference space in an atlas. So, why do we need all those complicated nomenclature and terms? So, because we really have to bridge a very large scale, and also we have to bring in different modalities. So here we can see different regions, one parcellation scheme. This is the BigBrain atlas, and this defines individual areas. Those areas do always have a variability. So, on the left, you see a maximum probability map where sharp borders are defined between adjacent areas. But normally, those maximum probability maps are built on probabilistic maps because each brain is individual. And if you really want to analyze it in detail, you have to include this variability, which is taken into account using those probabilistic maps. But of course, you also want to go into a higher detail, especially here if you're on the BigBrain Workshop, we also want to go into cellular details. And this is also the main topic of the second part of our workshop, so that we really can start from individual regions or points, going to a map or to a region, and from there on we can extract high-resolution data and also extract cellular data. And the last point are those multimodal features. which are also linked to different areas, like, for example, receptor densities or cell densities, but there are also connectivity data included, so there's really multimodal data available. It depends on your need, and it's an open system, so you can bring in your own data, but you can also use the data from other labs. As shortly mentioned at the beginning, so there are different atlases included. So we will here mainly focus on the human multi-level atlas, but there's also Vexome Space Red Atlas, there's Macaque Atlas included, and you can easily switch between those atlases, and you can also switch between the reference spaces, but this we will see in practical examples later on. This is a very short hands-on part just to see if everybody's able to open the browser and if everything works. So please try to open the link displayed here on the slide and see if it opens the web browser. For those who are not connected, here are the Wi-Fi passwords and network name. So please try it. And if you open the viewer the first time, you will be asked to do a tour. This only takes 1 or 2 minutes. Maybe start doing this tour. Simply click through those little comments. And if there are any problems, technical problems, please raise your hands and we can try to fix it at the very beginning so that we are not losing anybody at the beginning.
Just before you start, one remark that we forgot to make is we will be missing a presentation in the afternoon. So Dagmar Keinmüller, who was planned to give a presentation, cannot come because she fell sick, unfortunately. And we will therefore take a bit more time for the tutorials here. So we will take 2 hours instead of 90 minutes, which is good because we can go a bit slower. And if we don't need to go a bit slower, we will have time to look into a few more things with you, depending on what you find most interesting. So just that you are aware, also the people who follow this online, we are planning the tutorial for 2 hours in total, so until 3:30, right? Have to check.
2 hours.
Yeah.
Yeah, sure.
I could also put the link in the chat.
The link is also listed on this short printout, the Atlas Viewer link. So it looks a bit different, but at the end there's one link called Atlas Viewer, which brings you to the same web page. So if you have opened the web page for the first time, you should see this. You should be asked to do the tour. If you have clicked somewhere and you have lost the tour, you can press the H key on your keyboard, which will bring you to this help screen here. And here you can start the tour again. You could also open the, this help panel in a new window, just in case you need it. So let's do the tour together. So please feel free to click, to use the tool, to play with it. What the tour asks you, so at the beginning, as I said, there are different atlases included. So here at the left lower bottom of the screen, you have breadcrumb navigation, which gives you access to the different atlases, which is the left part here of the navigation. Here you can switch the reference spaces, and here you can switch the parcellation schemes. So those are exactly the wordings we have just introduced on the first slide. So now we are here, it's a human atlas, and we are in the MNI 152 template space. This is the reference space, and all those colorful patches here belong to the BigBrain parcellation, which are shown by default at the beginning. So if you want to change it, simply click it, select the atlas you would like to work with, and it will be loaded. And there's also one feature which is quite helpful if you really want to work with your own data. You can drag and drop your own nifties. So therefore you should have your nifty on your PC somehow, somewhere. I only do have a BigBrain nifty, that's why it won't work here at this point. But so make sure you are in the correct reference space if you want to drag and drop something. So if you have a nifty file in the MNI 152 reference space, you could simply click it and drag and drop it here, and it will be displayed in the viewer, and you can see it with the context of the atlas parcellation and the reference space in the background. But you have to make sure that the reference space is correct. This is only an overview, so you have, it's in 3D volume, so the BigBrain Atlas and the template, of course, also is in volumetric data set, so all those 3 panels are navigated together, they are linked. Here you have those cutting directions, the frontal, the sagittal, and the horizontal one. Here you have the 3-dimensional reconstruction of the surface, including the labels here inside. In some of the reference spaces, you also have the maps on the surface displayed. It depends a bit on the data set and how deep it is correlated in the system here. For each of those panels here, you can zoom in, zoom out. You can do it with your mouse wheel, whatever you would like to. And you can also increase it if you only if you're only interested in one cutting direction. Then you can go to the specific cutting direction. Here's this cross in the right corner. You can come back to the original view. This is also something which is quite helpful. It's often the case that you read in publication and they report functional activation at a specific coordinate, or you have a coordinate in a reference space. And if you really want to navigate, you want to know what region is located at this specific area, at this specific coordinate, then you can simply type it in here. The X, Y, and Z coordinate, since it's a 3-dimensional coordinate, you need 3, of course, and then you will directly be directed to the coordinate, and you will also see an assignment. So if at this specific coordinate you have entered a label was assigned to the reference space, you will also see here an assignment with the probabilities of being a specific cytoarchitectonic area at this position. Use the quick search for, okay. Wait a second. So there are different ways to select areas and to navigate within the browser, within the Explorer. So first of all, of course, you can use your mouse simply by hovering over a specific area. You will get a text field here showing you the area name at a specific localization. If you already know the area you are looking for, you could also directly type it here. For example, 4P is one of the regions of the primary motor cortex. So you could search for this area, and it will directly bring you at a specific position. So here you can see area 4P, and it will also navigate you to the centroid position of the specific area. If you have lost the position or you do not see the map, then maybe you have to click again the centroid button here, which will bring you back to the centroid of the selected area. There are also different tools which are already included here in the Atlas viewer. For example, you can do some annotations. This will be shown by Timo later on. SIIBRA UJEX is a tool which gives you access to genetic data provided by the Allen Brain Institute. So there you can do a gene expression differential comparison between the gene expression values of different areas, also directly here live in the browser. And of course, screenshots and something like this. Screenshots might be helpful if you want to share data with your colleagues. But what is also something specific about the SIIBRA Explorer, you can always restore the view you are seeing here in the viewer completely if you copy-paste the link. So all information, so where we have moved and at what position we are at the moment, the selected atlas, reference space, everything is stored here in this quite long URL. And you can copy-paste it and send it to a colleague. So if you, for example, have a specific position where you're interested in, or you have a question about a position, an anatomical position, you could simply copy-paste this URL, send it to your colleague, and discuss it, and at the same position. Okay. I hope you could reproduce those steps here on the tour. If not, as I said, you can always restore it by pressing H to get this help window here and take the tour again, just as a starting point to play with the viewer. Okay. I've talked already a bit about the Jülich Brain Atlas, and since this Jülich Brain Atlas is also deeply connected to the big brain itself, I also would like to give you short background information about this specific type of atlas and why we use it here. So the Jülich Brain Parcellation is built on microstructural differences between brain areas. So in order to create it, we have sectioned a collection of brains. We have about 26 in our institute, which are completely processed, which means they are digitized, they are reconstructed, and most of them are more or less completely annotated. If we section a brain, this results in about 7,500 sections. They are about 20 micrometers thin, so half of the thickness of a human hair, and they are digitized with 1 micrometer in plane resolution, which really gives access to microstructural details up to individual cells. And since it's based on microstructure, we are looking for differences between adjacent areas. So this is the most obvious border we have in the brain. Here on the right side, so you see the primary visual cortex. On the left side, the secondary visual cortex. And I hope you can see that here clearly more smaller cells in the in lamina 4 of the primary visual cortex compared to the secondary visual cortex. This is because our eyes do directly project here to the primary visual cortex, resulting in such a high cell density. But since, of course, it's not always that obvious, so this border you can even see without a microscope. If you know where to look for this border, you can see it by your naked eye on an object plate. But if you're going to higher associative areas, like those ones here, area 11 and FP1, they are located at the frontal pole of the human brain. The borders become much more difficult to detect. And therefore, we use methods to quantify borders and to quantify differences between areas. This is done by defining line profiles from the outer surface to the inner surface of the cortical ribbon. And those line profiles do correlate with the microstructure of the underlying cortical region. And here you can see that those peaks and the shape of the profiles are different. But since, of course, it's biological tissue, we have quite a lot of noise, and we cannot do it only on one profile. So we define profiles all along the cortical ribbon. And then we are using a sliding window approach, which moves along the cortical ribbon and compares groups of profiles. So we generate a feature vector, and then we compare the groups of feature vectors of adjacent groups against each other. And if we can see statistical differences at specific positions, then we take this as an observer-independent defined border, but this still has to be reproduced on different sections. So sometimes we have blood vessels or whatever causes some kinds of artifacts here. So we have to define borders, we have to detect those borders on a series of sections, which at the end is the strongest hint that there really is a border which can be reproduced. And once this is done, this is integrated into the atlas. At the very beginning, I've also talked about variability. So that's why we analyzed 10 individual brains. So here we can see 3 of them. In each of those individual brains, we map the specific areas. Here, the frontal pole areas. And once this mapping is done in the individual brains, those brains are transferred into a reference space. Here it's the Colin MNI 27 reference space. And also the annotations are transferred, of course, into the reference space. And once this is done, we can simply superimpose those annotations from the 10 individual brains and create such a probabilistic map. And those maps build the basis of the Jülich Brain Atlas. So red means here that the specific area, in this case FP1, was detected in 10 out of 10 brains at the specific positions. And here in the periphery, you can see that there are some blue and green colors, which means that there's a lower probability, and this is at the end the inter-individual variability, which is also included in those maps. And we think this is the best way to really to work with those maps in taking and including those probability information into your workflows, because, as I said, each brain is individual, and if you really apply sharp borders, you will definitely lose some information. Of course, it makes it a bit more complicated if you keep on all this probability information throughout your whole workflow, but at the end, the results should improve. So this was about the BigBrain Atlas as one parcellation scheme which is included in the SIIBRA Explorer. But as I said, it's an open system, so the whole eBrains infrastructure is built that you also can bring in your own data set. So it's specifically built for this. And this can be done in different ways. So for example, there's one tool, it's called Voluba, where you can anchor your own 3D volumes in high-resolution template spaces like the BigBrain space. And this can be done, for example, by image registration. So if you really, if you have a piece of tissue or volume of tissue where which was reconstructed, or you have 3D image acquisition build volume, then you can directly anchor this volume into the reference space. And by doing this, you can really use all the different modalities which are already included, because you bring it in the same reference space like all the other data sets are. And this gives you access to different resources which are included in the eBrains infrastructure. Sometimes you do not have such volumes or all the information around. So you can also do a more easy semantic tagging, so that you know that your data, your features were extracted, for example, in area HOC1 left, which is the primary visual cortex, here with the probabilistic maps displayed at the back of the brain. And this is, of course, the easiest way, because it does not give you really precise regional, precise information about the coordinate space, but at least you know it belongs to area HOC1. So there are different levels of anchoring your features inside the reference space. And another way, this is also the one which Timo will present later on in the SIIBRA Python part. If you already have coordinates, then you can really use those coordinates to anchor specific areas, or the other way around, you can use those coordinates to extract data which are already included. One thing which is already included in the SIIBRA Explorer is that you have different reference spaces. And normally, for those of you who have tried to register different brains into reference spaces, this can be quite complicated or challenging. Here, everything is already pre-computed, so if you really want to switch between the big brain into the MNI space, or also into the FS average, into the surface space, this is directly included in the SIIBRA Explorer. So if you remember this breadcrumb navigation, it's the lower left part. You can choose between the atlas and also between the reference space. And if you switch between the reference spaces, SIIBRA Python in the background will always try to keep your position as good as possible. So you can select position in the MNI space and switch to the BigBrain space, and SIIBRA will try to keep the position as good as possible and bring you to the same position. And by doing so, you have access to high-resolution data directly in the browser. One thing which is also very important, and for those of you who might have heard the workshop from SIIBRA this morning, all the data sets are really deeply curated. So they are not only labeled, they are really well described. So for the whole atlas, for the parcellation scheme, you can directly access, you can see this info button, it's the lower left. If you click to this button, this will open one of those slides here, give you the knowledge graph data set card for the specific parcellation scheme, here the BigBrain Atlas. This holds also true for specific areas. If you have selected an area, you can always click here on those links to these DOI links displayed on the left side in the browser, which will bring you to the dataset cards of the individual maps. So if you want to work with the datasets, you can cite them, but you get also information how those maps were created, who created it, when they were created, all the meta information you need if you really want to use them in your own publication. And this is also the same for the features. So for example, if you have selected the receptor densities or those multimodal features which are connected to individual areas, they have also their own dataset card. And everything is linked and tidied up and collected here in this workflow so that you really have access to all datasets which are included. Okay, now we really would like to work a bit with the viewer and to show the interactive navigation and also to work with the coordinates and to do annotations and extract information at specific locations. This will be shown by Timo.
All right, so the bigger picture for this course is, since the course is titled Working with Quantitative Cell Densities, you might have been wondering what do these things have to do with cortical cell densities. The bigger framing is that using the SIIBRA tool suite, we can model a workflow where you find the location in the brain and then you search for relevant measurements, relevant data sets. That's what Sebastian already outlined. So this has, of course, two bigger parts. First of all, to identify what you mean by a region of interest. And I'm just getting signs that I have to move closer to the microphone, sorry. So you first of all have to specify a region of interest in some form. That's what I want to show now. And then you need to understand how you can use that then to find data features linked to the atlases. So let me just give you a brief overview of these things. Some of them have already been mentioned by Sebastian. Of course, the very obvious way of finding a region of interest when you use atlases is to select a brain region. Yeah? So whenever we click here on a region, you see that the viewer loads the probability map here in that case because it's available for this atlas, and it opens a side panel where this region is selected. And of course, this region has a centroid. We can actually zoom in to the region, and you see right away here, maybe I make that a bit smaller, you see right away here a panel of features that have been linked to that particular brain area. I will get to this later again. This is a very obvious way, yeah? And I can, of course, instead of just navigating around, I can also search for regions. So here's this, the top left of the window, there's this magnifying glass, and here I can also write the name of a region, yeah, 4P, click on it, and this, of course, also leads to the selection of a brain region. Yes, and you can, of course, also, by clicking here on the very left, you can also see the full region tree of the currently selected parcellation and browse through the hierarchy to end up doing the same, yeah, selecting a brain region. So when you click then on one here, this is selected and you move to the position. This is selecting a brain area, and this gives you two forms of locations. One is the name of the brain region, and this is exactly a semantic entity or basically metadata if you want. But the other is, since we have selected here a particular space, a particular template, and we have access to the parcellation map, of course we also get the segmentation mask, which has a bounding box. So I also have something, a spatial element, right? For example, the bounding box, or what I'm looking at. And these spatial elements, of course, we can always deal with them independently of brain areas as well. So I can of course just move around here, zoom in, and find a certain location And this location is at that stage just encoded by the viewer, and it means it is also encoded in the URL. So if we would copy the current URL, I can just do this here. I'm usually not a Windows user, you see already how, but that makes me slower, and I heard it is good to be slow in a tutorial. So if I open a new tab now, and I just paste here that same URL, it takes me to the same location, yeah? Because the position and the zoom level and the orientation is simply encoded in the URL. And that means the URL is already a specification of a location in the brain, because it encodes the current position, zoom, level, orientation, which is also a bounding box in a way. And I will show you later on how we can work with SIIBRA in Python. And in fact, you can use the URL. You could actually copy over the URL, take it over to a Python notebook, and in SIIBRA Python, it can parse the URL and it knows where that is, in which space, and so on and so forth, so that you can continue working with it even in a programmatic fashion. But of course, there are different ways to specifying coordinates. One has already been shown by Sebastian. If I click here on the location bar, I can simply enter a location. For example, I can enter the MNI coordinate on the Mac. I could just double-click. 1, 2, 3, and then I select this point, and then it moves there. I'm not sure if some of you know what 1, 2, 3 in the MNI space really means, but you can see here already, I have now selected this region, and it has already shown me here an overview which brain regions are present with a certain probability at that position. So of course you can just take a note of the MNI coordinate. That's also a way of specifying a location. It is probably very trivial things I'm telling you now. If you want to do something more, let's say, more well formalized, there are ways to do so. So first of all, the current view, the current URL, instead of just copying it, you can also share it. So you can, again, go to the panel here, and there's a little share button, and you can even create here custom URLs with specific names. So if you're logged in with an eBrains account, you can here create a custom URL, give it a name, and this should be persistent, and you can share it or use it in a paper. But there is also an annotation mode. For that, let me just move to the BigBrain space, because it's a bit more interesting there. So if I switch over to the BigBrain, I'm not sure if Sebastian mentioned that already. The system will then try to preserve the current view. So if we switch to the BigBrain, that's a different coordinate space. But actually in the background, the system is already using nonlinear transformations to translate the position and the zoom level in an appropriate fashion. So if I go back here and then zoom in even further, and I could maybe disable the parcellation here at this stage. And then you see, of course, in the MNI template, when you zoom in very much, it's quite blurry and doesn't have a lot of detail. But we can use this quite nicely to then switch over to the BigBrain space here. And we see a zoomed-in view in the BigBrain at approximately the same location. Of course, that cannot be exactly the same, because this is an individual subject. The other one was an average subject. You're probably aware of all these problems. But it should be pretty good. So now in the BigBrain space here, it's actually quite interesting to find specific coordinates. And when we use here the plugins and tools on the top right, not the download button, which I now clicked, there's an annotation mode. This opens a small side panel. It is very basic, but it is very helpful. So here you can select different tools on the left to annotate structures. This is not a fully fledged 3D annotation tool, so you cannot use that to mask 3D regions in the 3D volume or something. It's much more basic than that. You basically draw on what you see, but nevertheless, you can use it to draw points or 2D lines or even polylines. In a very simple case for point, I just select here the point selection tool. I find here an interesting cell, click on it, and there is even already one. Sebastian already has here a named annotation. I just created another one. Yeah, so I can give it a name here. Timo's cool cell. And there we go, and now Sebastian has it in his browser memory. And from here, you have actually the possibility to copy the specification of this annotated point. So here's a button, export the annotation. At the top, here's also a button to export all your annotations. If I click on that, you see here already a JSON description of the structure. Yeah, and if I just copy this over, and am I allowed to open the text editor here, Sebastian? Wie geht denn das unter Windows? Text Editor. Gibt es sowas unter Windows? Notepad, ja. Very good. So if I copy paste this here, you actually can see this is a pretty lengthy text file to specify a single 3D point. But what you can see here is it follows the open minds schema that Liuba Zehl has described in the morning. And it does not only just specify the coordinates, but it provides also information in which reference space this was annotated. So this specification is really sufficient in a batch processing mode to know exactly what we talk about. Yeah? This is an exact specification, which reference space, which coordinate that can be processed. And we will see later on how we can use such a description here to actually move over from the interactive workflow that we have here in the browser now into a programmatic workflow in Python. We will do this in a second. Yeah, so maybe I think that some of you are clicking around while we're presenting. Some of you are just listening. That's both fine. Just having a quick look. If there's any questions. So this annotation mode, as I said, it is basic, but it is quite useful. You can also use it here with this multi-tool to click more extensive structures, a triangle or whatever. You can actually even click across different sections and put the polyline into full 3D, but it's not a tool to annotate surfaces, as I already said. All right. I move on with finding data, right? So you saw already that I started by explaining we go from specifying a location to finding data. Now you saw several ways of specifying a location. The simplest one was a brain region, and you have already seen, if I select the brain region, the side panel will open. There's a list of features. I can also do that here in the BigBrain space. Although this BigBrain space is not yet fully mapped, you see that there are quite some region mapped here, and actually there's a big update coming. I'm not sure if Kim Lotman is here, but she worked a lot with several other people to get many more BigBrain maps in, and I think she has a poster to present on that topic. But some areas are linked. Here's a primary visual cortex. Simple example, if I click that, the same happens again. I see here in the side panel the brain region with all the metadata. I see again that this brain region and the map we are seeing has a DOI. Yeah? So when I click on this DOI, it takes me to the knowledge graph. And you see there's a data set of this particular high-resolution map in the BigBrain space. It has a DOI, it has a description, and so on. And I find here this panel with the features. So this shows me data features in simple words data sets that were explicitly linked to area HOC1 or something related. They could also be linked to a sub-region or to parent region. Then they would also be listed. So what you can see here now that is, for example, a quite lengthy list of brain sections, of histological brain sections that overlap with area HOC1. You see that here. Yeah? You see that there's some information about nerve fibers. We have here some whole brain volumes that are of course related. There is some molecular information, and this is prominently receptor density measurements. And if we take a high-level overview of these data features, you can usually say some of them are images or measurements that live in a coordinate space. Some of them are tabular data. So it's just more or less is an Excel file. Yeah? So these receptor density profiles here, for example, this is tabular data. If I just click one of them, then SIIBRA will actually load it. This data set has again a DOI, and SIIBRA can also show it. Yeah? So we see here that's a cortical profile of densities along the cortical depth of the M1 receptor that has been measured in the primary visual cortex. So this was a CSV file that was taken from eBrains and is just visualized here. And the original location can be found because, again, we see here a DOI, we see a data description, and if I click on the DOI, you can actually see that's a data set here on the knowledge graph, and you could also download it here. SIIBRA just made the job a little bit simpler and allowed you to do this while you are browsing the 3D anatomy, but of course you could also basically find the data set here and download the same measurements here. For the case of data that lives in the coordinate space, for example, images that have a bounding box in the coordinate space, we do not have to specify a region selection. We can really search with coordinates and spaces, and that's why we can find them while we are here browsing the 3D space. Yeah? So you might have noticed that there's a little number here on the top while I'm browsing. It currently says here 20. That means it has found 20 typically images that are actually anchored to the current view. So as I zoom in and move around, the number changes. So you see now it's 11. It's less than before because I have zoomed in. Can zoom in even further. It says probably now 5. Yeah, 5. And if I open that here, I see now a list of data features that are anchored to the current view, not related to a brain region as previously, because I have not selected a brain region anymore. They're just related to the current view. And here I see again some brain sections. Now that I opened the side panel, it even shows me where these brain sections are. So you can see here the yellow stripes. These are coronal sections that have been anchored to the big brain space. In fact, these are actually big brain sections, but full resolution 1 micron sections. So I can click one here. And again, in the side panel, it shows me the information about the data set. It shows me the UI of the published data set. And it sees if it knows enough about the data to load data. In that case, it does. It knows how to load such a 1 micron resolution brain section. And in fact, it has already been loaded and is already shown here in the coronal plane. You might have noticed that the contrast here is now a bit higher. And indeed, when we zoom in now, yeah, you see that the sagittal and axial sections still have a 20 micron resolution, so somewhat limited as I zoom in. But in the coronal plane, I have now a full resolution 1 micron section selected, which has a much higher resolution. Yeah? And I can again maximize the view, and I see individual cells.
You can also see the border we have seen that's at the beginning. So since we are here at the primary visual cortex, if you zoom out a bit, you can, we have here the maps.
Yeah.
Go to the right side.
We can actually double check the maps. So you see here's the map secondary visual cortex primary. So somewhere here, or maybe here at the top, it's more interesting. Here is a transition between the primary and secondary. I disable the map. Let's zoom in, whether we can see something there. We have the experts in the room.
And those exactly the positions you have, not exactly the same position, but you can see the higher cell density in the lower primary visual cortex and the lower cell density in the upper secondary visual cortex. And you can zoom in up to 1 micrometer directly in the browser. Yeah. So this is really 1 micrometer information. And each of those images is about 10 gigabytes in size. And there are currently, I think, about 150 sections are linked and registered here in the Explorer. And you can directly access them using SIIBRA Explorer also, or using the SIIBRA Python library, which will be shown later on.
Okay. Very good question.
Can you hear me? Yes, okay. Yeah, so I work with surfaces. Can I just stick in the vertex coordinates into SIIBRA or whatever is viewer and extract data at those coordinates?
You can do that with the Python client. Yeah, we will show that now in a minute. You can also, of course, do it here, but it depends, of course, what your coordinate is. If you have an MNI coordinate, you could do that in the MNI space, then switch over to the BigBrain and look. What I should say is that as of now, we have a set of more than 100 such full resolution sections in the BigBrain space, but not every section. Yeah, for a couple of reasons, not every section has a right quality and so on, so there are a couple of issues there. So it means you will not find the 1 micron data at every arbitrary position, yeah? But you usually find something close by. Yeah? And we are constantly enriching this with more data, of course.
Right. But I guess the Python workflow then would tell me which is if I'm hitting or very close to the section, yes. And I could extract the data from there.
Yes.
Okay.
In general, both systems are directly connected. So under the hood here is also SIIBRA Python working, and all the things we can visualize here in the browser. This is thought to be the very easy starting point if you really want to play with the data, visualize it. If you really want to work with the data, use it in a programmatic way, then you should use SIIBRA Python part. But everything you can see here can also be done in SIIBRA Python, including high resolution data up to 1 micrometer with coordinates, extraction, everything.
We will do that actually now.
Okay, nice. Yeah. I'll try it out.
So if we take a step back at this stage, what we have seen is we have interactively in 3D identified certain locations, regions of interest in the brain, and we have seen that for these regions of interest we can find additional data, and this data always has a link to some cloud repository. In our case it's often eBrains, but not necessarily only eBrains, where you can find the data. Some of this data, SIIBRA itself knows the data format and it can even load it already. That's not always the case. Sometimes you just get a link to a data set and SIIBRA does not know how to load that for you, yeah? But you still get a link. Now, if you want to have that reproducible, that's tricky, right? That is a fully interactive workflow. Yeah? What you could do is you could copy the URLs. Store them in a document for you, in your lab notebook, or you could, as I have shown you, you could annotate certain things, store the JSON files. So there is a way to preserve what you are doing, but of course that is not it's not programmatic, yeah? It would require a lot of discipline for yourself to make this reproducible. So such an online viewer, it is definitely more an explorative tool. That's also why we call it SIIBRA Explorer, than a tool to carry out your study, to do a reproducible study. And that is why we have modeled it in such a way that this all can be done in Python. And we will step over to this now. I will just show you 1 or 2 more slides at this stage. So again, what we have done here is we were browsing at 1 millimeter. We were selecting certain regions of interest. We were switching to the BigBrain space, zooming in, finding high resolution images as an example, ending up at a very precise location. If you were to do that same thing in Python, that would actually be very easy. This is a figure from a preprint we have on bioRxiv at the moment, and there is a second figure that shows the same thing in Python, and it would really look only like this. It's so that is of course a very simple example, but what you're doing here is so first of all, who knows basically how to program Python? I mean, not professional software development, yeah. So you can read some lines of Python, right? For the others, ask if you don't understand, because this here does not require a lot on deep Python understanding. The message at this stage is just that you can write down basically the instructions to carry out something instead of doing it yourself with your mouse. So what you do is you import the library. In that case, we have selected here a parcellation from SIIBRA. So the Jülich probability maps, we have specified, as it was the same in the browser, we have specified the parcellation itself with the version in that case. We have specified the space and the type of the map, which when we say statistical, we get the probability maps. We could also say labeled, then we would get a labeled map, a simpler representation. But here we use the probabilistic maps. So this is a set of maps in a particular space. From this set of maps, we now extract an actual image volume referring to one particular region. In this case here, I selected 4P right. Yeah? So I retrieve the volume for this particular region. So I get a region map. This is still more than a nifty file. This is not a nifty file. This is a structure that knows where to get the nifty file, and it knows a few things about the nifty file. For example, that it represents that brain region, that it is actually defined in the MNI space, and so on. But under the hood, it then actually knows how to provide you the nifty file. So in SIIBRA, a brain volume basically means you have a description what type of volume it is, in which space it is, and where to get it from. Could also be a mesh, but now we have a volume which is an MNI, a nifty file, because we are here in the MNI space. And then we can use this brain volume that contains a map in the MNI space for area 4P right to run a feature query. And a feature query always looks the same. We have a query concept. In that case, it's a brain volume with a statistical map, as you see it here in the figure. And then we define a type of features we are looking for. And now we want to actually get access to a 1 micron patch in the BigBrain space. This is more than just a 1 micron section. We actually want to sample a cortical patch in upright position, which is a rather involved process that is implemented in SIIBRA. And then we get a couple of features, that is a couple of 1 micron cortical patches relevant for the selected area here. And we just select one of them. So what that looks like and what happens under the hood is we have this map in MNI space defined at the millimeter resolution. This map is used by SIIBRA to search through the available brain sections, which we have just seen in the browser ourselves. So it finds one, it is here indicated with the dashed line, which sits at the maximum probability here. It finds actually several ones. This is just one now illustrated. And inside that brain section, again using the probability map, it actually knows where to look, right? Not the whole brain section is relevant for area 4P right. It is only a subpart of the whole section. So only that part here is actually relevant. Well, maybe a bit larger. This is actually, so it finds a region of interest to search in the brain area. And then once it's there in the brain section in a zoomed in region, it actually internally loads cortical layer maps to understand the orientation in the cortex. So where is outside, where is inside, where is actually the cortex? Because if I want to have a correctly sampled patch in the cortex, I need to know where's the outside of the brain, where's the inside, and so on. So it uses the cortical layer maps to actually identify a profile going along the cortex, understand what the thickness of the cortex is to define the size of the patch, and then it actually extracts such a 1 micron resolution cortical image patch that is very likely to belong to that brain region that we used as a query. So that's 3 lines of code here, but of course a lot of stuff happening in the background, a lot of interactions with data that lives in different coordinate spaces and so on. Yeah. And the good thing about that is this is reproducible. Yeah? This is 3 lines of code. If you keep track of the software version that you were using, this should actually always produce you the same list of patches. Although what you get in the end is not an image that is somewhere out there. The images that we have stored in the system are these whole brain sections, and they are huge. Yeah? If you want to download them, even one section is usually 20, 25 gigabytes. So that is usually not something you want to interact with if you're interested in regional information. Here you get a patch extracted from that section, but by storing this piece of code, you have a reproducible snippet that describes how you actually ended up with that. And it also encodes exactly the coordinates and the bounding box of the data. So you could really describe at any time where this was taken from. You know the brain section, you know the DOI of the data set, the bounding box, the resolution, everything is there. Encoded actually in this feature here. And I would like to go through this process with you. We talked about quantitative densities. What we have here is an image. That's not a quantitative density or anything like that. But you could imagine that if you use this and you run an AI model to detect the cells, you would actually, from here, end up in instances of cells which you could translate into densities. And I also want to do that with you today. Just before we go into the Python ourselves, a little, what is this? Is this called a teaser? So in an upcoming version of the SIIBRA Explorer, we also want to enable to detect cells right here in the browser. This is actually already implemented, but we have not yet released it because it's computationally intensive, and we have to see how we do the practical setup if we open this for more users. But actually in the future, you will also here in the viewer have the possibility to select an AI model that can detect cells from a list of models that we know that can operate on such data. And when you zoom in sufficiently close, it will show you live segmentations of the data as you move along. That's not yet released, but that will come. Now I want to do Python with you to do the same thing, basically what I've shown you. And for that, we have also prepared a link, which is this one here. It's a tiny URL. I think it was already probably pasted by Susanne also in the chat for the online participants. You could also take a snapshot of the QR code or just write down this number in your browser. This should open an interpreter with a prepared Python notebook for you. Please do not run the notebook yet. All of you. Let me make a few comments first. But you can safely already type in the URL and open it. I keep it open. So these notebooks will then be running on a cloud service that is called MyBinder, and it doesn't have super many resources. So I tried it myself, and I could run the notebook, but I don't know what happens if many of us run it now. We will see. I have the notebook also on my screen, so we can also follow along. But please don't run the whole notebook yet, because some of the code in the notebook is requires a lot of memory, and it might not work, and I will give you some instructions as we go along. So just open it. You can run the first 1 or 2 cells, but we can also do it together. Did everyone have
questions or problems?
Yeah, just ask. We have half an hour more today, so we have lots of time for you to ask questions. Otherwise, I will just tell you more stuff. So it's your choice. All right, did everyone have a chance to type in the URL? And is there a notebook opening for you? Is it taking long, or do you see the notebook already? Yeah, okay, good. Then I need to take my machine now, I think. Yeah, but I want to do my local version. So at least one of us has a local version.
Okay.
Very easy, okay. So I have the same notebook here. For me, it's running on my local machine, though. And you can, of course, download it and run it on your own machine if you have Jupyter notebooks running there. I just wanted to avoid in a short time here that we do installation sessions for Python, for Python packages. But it's usually not difficult to install SIIBRA and the dependencies there. And we will also provide you with links to documentation where it's described how to do that. We just wanted to avoid doing it here. Yeah. So what you see should be the same here. So in the first cell, we just import SIIBRA, of course. And here in these notebooks, for those of you who don't use them, basically these notebooks is a sequence of cells, and these cells can be executed. You can execute them with the play button here. Please don't use the double one, because it runs the whole notebook, but this one, or Control Enter. When you execute a cell with Python instructions, it executes the code. When you execute a cell with text, it renders the text. So we basically can, with the play button, you can go cell by cell, and it will either show you the formatted text or run the code. So what's happening here in this second cell is I'm I have copied in here the JSON string of a position in the BigBrain space that I annotated in the SIIBRA Explorer, what we just did 10 minutes ago. Yeah? So I used the annotation mode, I clicked the point, and I copied there the JSON, which is that one. So I can now simply say, and you could very easily for yourself modify this now. So you can annotate a different point if you wish in SIIBRA Explorer, copy it over here. So what we are doing here is we just paste it in as a string and tell SIIBRA, please create an object from that JSON file. And this object is a point. So if I show that point now here, you see what SIIBRA is telling me, that's a point at this particular position, and it is sitting in that space with this ID, which is the BigBrain space. What I want to do now is, other than the slide I've shown you where we started from a brain region to find a cortical patch with cell densities, I want to start from this coordinate. And at the moment, SIIBRA, for sampling these patches, it assumes that we have a nifty volume that encodes a region of interest. So what I do here now, and in a future version you would probably not have to do that, is I build a volume from that point. Yeah? Basically, I warp the point as a point cloud and provided here a certain uncertainty, 0.3 millimeters. I just tell it this point has a certain uncertainty, a certain radius around it of tolerance. And then from that point cloud, I ask SIIBRA to build a volume. So what it then does, it basically builds an image, a nifty image with a Gaussian blob centered at that point with a standard deviation of 0.3 millimeters. So I have a kind of a very tiny map of my region of interest, which was just a point originally. Yeah. And this I can use in the same fashion as I have previously used the probability map of area 4P to go into the process. Here in the next cell, I'm just showing that. So we can see here a plot of a lower resolution version of BigBrain, which had been downloaded, and superimposed I show this volume, which shows you this little blob around the coordinate. To show the BigBrain, you also see here that's not difficult. Yeah, I just tell SIIBRA get the template BigBrain, fetch the image data, and when I say fetch, what it usually does is it looks how large is the data on the other end. In that case, how large is this image volume? And the BigBrain image volume is very large. I don't know exactly, but maybe going in the order of a terabyte. So it will, of course, not download the terabyte of BigBrain. So what it then does is it downloads it and provides me a copy with a reduced resolution. But it tells me so, and the resolution is also, of course, encoded in the nifty file that I get. And I can always provide parameters here to specifically ask for higher resolutions. Here I'm not interested in a high resolution because I just want to use the BigBrain to see where my query volume is. And then I use just the plotting functions from nylearn, which are very nice, and everything that SIIBRA returns as an image is typically just a nifty image object, which nylearn can work with very nicely. So we can just plot that on top, and you see that this query volume is defined in the BigBrain space, and the point is here where I annotated it. So now to run the query, you remember that from our slides. If we have a look again, where's the slide? That's my computer now. So we have been looking at this here, and you remember that there was just a single line here, features.get. I provide the region map as a concept, query concept, and then I specify the type of feature I want to do. Same happens here in the notebook. Yeah? I put now my query volume as a concept, and I select as a modality BigBrain 1 micron patches. If you run that cell, it will very likely take a little bit. Because what's happening now is, did someone run the cell already? Yeah? I'm not sure if you... So this will take a little bit because SIIBRA is built in such a way that it loads only lazily. It loads only lazily. That means it will only retrieve things from the online resource once they are needed. Since we started this notebook in a virtual machine that's out there in the cloud, which has never previously used SIIBRA, it means it never had to load anything. Now is the first time. Now we look for these 1 micron sections, and now suddenly SIIBRA needs to know about the brain regions, it needs to know about available brain maps. You know, in the background it uses the cortical layer maps, so it needs a lot of stuff. And now it will, if you run that for the first time, it will now suddenly retrieve these things. So this will probably run a minute or so, this cell, and you will see that how it's loading different things to run this query. The second time you will run the same cell on the same machine, it will be very fast because the stuff that it often needs, it will just keep it in a cache. Yeah? But the first time, you will probably see it loading maps and stuff now. And I think it will probably take a minute. But you will end up here with a list. If you took the same point, in my case, it was a list of 3 cortical image patches, which it could actually identify in that region of interest. And it makes some selections, right? It only takes positions which have a high probability according to your map. It only takes positions where a 1 micron section is actually located and where the cortex sits somewhat nicely. So it checks a few things. Therefore, it doesn't find infinitely many good positions. Can you still follow along? I'm usually told after these sessions that I'm going too fast. That's why you probably realize I'm looking a lot at your faces. Can you follow along? Is it still fine? Or did I lose you? All right. Good. So did anyone have success running the cell? Retrieved some features? Or are you anyway just looking here? Just looking here, okay. So what we have then, a couple of features, and what I do here is I just select the first of them. And as I said, everything is lazy in SIIBRA. So we have not yet at that stage actually downloaded the 1 micron data. What we have from SIIBRA is actually a protocol how we could get it once we would like to have it. And at the moment where I say fetch, it is now actually executing that protocol. Yeah? So this object patch, it actually encodes everything. It encodes which brain section we have, what is the bounding box, what needs to be done, what is the DOI of this bounding box, and so on and so forth. And now when I say fetch, it actually says, okay, now I actually go and get the pixels for you. So that also takes a little bit if you're on a slow network connection, because this will now actually produce a 1 micron resolution image, which probably has a resolution of, I don't know, 2000 times 3000 pixels or something. Even if it looks tiny, it's a large image. So this also takes a little bit, but not forever. I have already done this before the session, so I don't have to execute it. And now I have a nifty image, which is a 2D image, but which sits in the BigBrain space. So it has the spatial metadata in the object to position it exactly in the BigBrain space. And this has 2 consequences. One is I can still plot it on top of the BigBrain, although it's such a huge resolution difference and everything. And I can also use its bounding box to get other image data at the same position. For example, I might be interested in the cortical layer mask for that image patch. And in BigBrain, we have cortical layer masks, the ones from Konrad Wachsdühl and colleagues. You saw them already. So what I can do, and what's happening here in the next cell, is I can actually get the cortical layer maps in BigBrain space, and then I can fetch the cortical layer from the cortical layer maps in the left hemisphere using the bounding box of that patch of my 1 micron image. So I can use that bounding box, can reuse it, and now fetch the cortical layer maps that correspond exactly to this tiny piece of image, yeah, to get a layer mask. And all of this sits properly in the BigBrain space. So what I do here in the next cell is just show that. Yeah? I first plot the BigBrain image in 3D, and I plot my 1 micron patch on top of the image, and it is so tiny that I almost cannot see it. Yeah? It's here. It sits at the expected position, but it's of course tiny. You can see it sits in the BigBrain space. Now I can also plot
without a background image, just the patch. And what you can see is it has this orientation because we have found it here in the sulcus, so it sits rotated in the BigBrain space, but it has the full resolution. And then the extracted layer mask also sits accordingly, right? So you can see here that our patch actually contains a little bit of layer 1 and 2 on the second, on the other side of the sulcus, and we also have here, of course, the cortical layer map labels on the other side. But for our pixels in the 1 micron patch, we have here now a mask of cortical layers. What you should know is that the cortical layer maps by Konrad Wachstum, they have been computed at the 20 micrometer model, and they also use quite some regularization. So if you go to that level of precision, if you go into the 1 micron data and retrieve the cortical layer masks, they may not always sit exactly to the micrometer. Yeah? Sometimes they are really a bit off, sometimes they are they sit very well, but if you use that, you should always double check with your own eye whether it makes sense. Sometimes it's shifted a little bit if you go to that level of detail. Yeah. The idea with the notebook is just to show you that you can all do this in a programmatic fashion. Yeah. So what we have here now is currently we are dealing here with 3 nifty image objects. One is the BigBrain at the lower resolution, one is our 1 micron image patch at the 1 micrometer resolution, and the other one is the corresponding cortical layer mask. It is a bit lower resolution. I don't know exactly what we did, but that's definitely not at 1 micrometer. Yeah. So we have 3 images, but they are all defined in the BigBrain space. What we can do now is we can use, actually, if you look at the voxels of this patch, if you only look at the voxels, you will see it's upright, it's correctly oriented. So if you don't use a nifty image viewer for this, but just look at the array, you will actually see that it's rotated in a way that layer 1 is on top. Yeah, I didn't do that here. I can actually do that to show you. So we have here the patch image. That's what we used, and you see that it's a nifty image. Yeah, and I can say get F data. This actually, that's not SIIBRA. That's just HIBALL. And you see now we have a numerical array. What is the shape of that array? It is 3D, of course, but it is in fact a 2D image. So I can say squeeze to get a 2D image. And then I really have just an image array, which I can, of course, also plot as an image. But now I have lost all the metadata, you know, that it sits in the BigBrain space and so on. But if I now say show this array with a grayscale map, yeah, you see that the actual array is already rotated upside down. Yeah. But if I use the nifty image, since this defines it sits in the BigBrain space, it is rotated because that's how it was localized in the BigBrain space. So we know where it sits, we know its rotation, but the actual array that we get is already resampled into a nice upright position as you would expect it for a cortical patch. Okay, now I promised you we will try together to also get some cells. And for that, we can use an AI model that we have published a few years ago, the contour proposal networks by Eric Upschulte from our team in Jülich. It's actually this one. There's a publication here, and this was also got a very good ranking up among the top 3 in a challenge at NeurIPS. So it works quite well on that data. This model is available on a page that's called celldetection.org, not detection, celldetection.org. And if you go there, you can already see examples. But what you also see there's different model names, different versions of trained models on different training data sets. And you can see here there are different ways to install it with Docker and so on. But what you also can see is that it is available on Hugging Face. That's a cloud website that actually hosts AI models, and you can upload data there and apply the AI models to the data there on that web page. And this is for our purpose very nice, because then we use the compute resources there, and we don't need to use the compute resources here in our tiny notebook. And that's why we're using that here. So there are only a few things to do. We already have our image patch. The problem is this AI model expects an RGB image, a 3-channel image, color image. So we have to convert the patch into a fake 3-channel image, but that is actually very easy. So I just extract the data here. I take only a small part of the image because I'm running this in a small notebook. I'm not taking the whole image. I divide by 2 to the power of 16 to reduce it to 8-bit. And then I just stack it here into three channels. So I get a 2D image with 3 channels. This is just because the AI model expects it. And this is a little, it looks a bit complicated, but just a small trick. I don't know the size limit, no. Actually, the model itself can, of course, handle large ones, but it depends on the server on which it is running, on the machine it's running. So what we do here in the variant 1 is we run the model on Hugging Face. There is a little package, a CRED.io client that you can use for that. We do that. Then we launch a client on in the domain of Eric Upschulte here for the cell detection packages. We save our image as a temporary image, and then we can just ask the client to predict for this temporary image with a particular model. We use this model here because I know it works quite well on this 1 micron BigBrain data. So if you run that, this should go pretty fast actually if you're still working in your online notebook. Because this machine, it either says you tried too many times, now you have to wait for 1 hour because you did too often, but if it accepts your request, it's actually pretty fast because it has GPUs and everything there. And then what you get as a return, as a result, is a couple of files. One of these files contains an image overlay, which you can show right away. But there's also some files with the detailed information about the contours of the cells and so on. Here I'm just showing the overlay image to show you how that worked, and that we have now actually a cell segmentation here. You see that's not the whole patch because here at this stage I just extracted a sub window, yeah, to keep things efficient. But the notebook also works for the whole patch. It just takes a little bit longer. So if you run this on your own machine, there's actually no need to cut anything down. It should work. Yeah. You could do the same thing without Hugging Face. So on Hugging Face, if you do that many times, it detects your IP address, and then it will say you did 5 predictions now, it's enough. But you can, of course, do it on your own machine. If you have a powerful machine, that's what I have here in the variant 2, which I'm not running here. You can actually import cell detection as its own package, and then you get the same result here. Here's the same model name. You load the model to your machine, and then you can run the prediction directly on your own computer, and then of course you have only the limits of your own hardware. This does not work in your notebook because fetching the model itself is already, I think, 800 megabytes or so, and then you get already into trouble with the online notebook. If you do it on your own machine, it should usually work if you have a decent workstation. And it should, of course, produce the same result.
Maybe for those of you who are not using those Python notebooks in their daily work, so Timo always said it's very easy to write those few lines of code. You can use this notebook and it will be there also for the next weeks and months, and you can simply change a few lines. And also at the very beginning, he started with downloading the annotations he made in the Zebra Explorer. You could simply take and coordinate. If you have a coordinate from your publication or from another publication, you want to know what's the cellular architecture, it's a specific position. You can simply define this coordinate at the very beginning of the notebook and extract cellular data for this specific coordinates you are interested in. So it's only an example how to get the 1 micrometer data, and all the rest is pre-configured. So you do not have to install Python. Everything is really here in the notebook. So just to make this clear, that it's really easy to use, and everything is already pre-configured.
Yeah, I can show that here. So there's a GitHub page, SIIBRA tutorials, where you find several of these tutorials, and you even find the link here again to launch them on Binder. binder. I actually don't know how it's. And you don't see our workshop today here because I created a new branch for that. So if you want to particularly do what we do today, you find here a branch, BigBrain Workshop. We might probably merge this into the main branch this week, but currently you would have to select this other branch, and there you actually see here BigBrain Workshop 2025. You could click here and you're back. Yeah? And you could, of course, also simply just find the notebook here and download it here. The other thing that Sebastian mentioned is the read the docs, zebrapython.readthedocs.io. There's documentation about Zebra Python, and the text and documentation we could spend more time writing more text, but the most important things here are examples. If you click, and these are quite comprehensive. Here in these examples, you find short code snippets for everything you have seen today and more. Yeah, so it starts from very simple ones. Just select how you select the parcellation. Yeah, so there is here a documented code snippet how you do that, and you can always download this. This is always a Jupyter notebook that works. Before we put it here on Read the Docs, it's always executed, so we know it works. And you find more advanced ones, multimodal data features. So here's connectivity matrices, whatnot. Here's also one about cell distributions. So when you go there, you find many examples. Usually they are short, and they have a lot of description what's happening, and you can download it and run it. All right. And this one here already brings me to the more or less the last thing I have on my normal schedule for today, cortical cell body distribution. So I showed you now a workflow, basically how you could yourself find the location in the brain, find the 1 micron section, extract the patch, run a cell segmentation there. And we also extracted the corresponding cortical layer maps. So you could actually produce statistics in that patch. How many cells did I find in a particular layer in that patch? And from that, you could, of course, compute what was the density of cells per layer or something like that. Yeah. Of course, we have done that ourselves already. And we spent quite some effort in doing that well. And that is why you also find some pre-published data sets with such statistics of cell densities. And there's actually more to come soon, but there are already such data sets out there. And in the last, here's my notebook again. At the end of the notebook, you see here that there is a feature type that's called layer-wise cell density. And if you search for layer-wise cell densities, these are basically tabular data linked to brain regions, so to cytoarchitectonic regions. So if I search by the Jülich brain cytoarchitectonic parcellation for this data type, I get a list of data features of brain regions from the Jülich Atlas, currently 12 of them, which contain such densities. So if you look at this, at the data, this is actually then a data frame. And what it contains is for one particular region, one particular brain region, we have here in different sections and different image patches sampled from these sections. You see that here. In a particular region, here's motor area 4A. We have counted cells in different cortical layers. We have computed their average cell sizes and densities. Yeah. So you see here actually, I think this is in 1, 2, I think it should be about 10 different patches in different sections where we have provided such measurements. So you can also, instead of looking at the data, you can plot this feature, and we see the plot further below. It shows a box plot here where you see we have layers 1, 2, 3, 4, 5, 6, and for each layer there is a point which stems from a different cortical image patch that was sampled, and we can see here the number of cells that were detected per 0.1 cubic millimeter here. Yeah. So we can see here in the motor cortex there was a pronounced density in layer 2, for example. Sebastian could say more about that. I'm not an anatomist myself. But you see a very clear signature here, yeah? How in different layers the cell density is, of course, a different one. And you could, from the same data, also produce such a statistic that considers also the area of the cells, not only the presence of the cells. Okay, this type of data, there is a much bigger data set coming that we are preparing now. So we have a data set in preparation that contains almost 1,000 such patches. And the cortical layers that were used are all annotated by experts. So they're also very, very precise. This will be coming. But I don't want to promise a date, but it is in the making. So it will not be in the very far future. All right, I need to look at the clock. So how much time do we know? We still have half an hour left, okay. How do we use this half an hour? I can easily talk for half an hour more about certain things, I think, probably. But the best idea would probably to use some of your questions and go into what you find most interesting, probably. So do you have some questions or some things that you observed here? Yeah.
Okay. Thank you for the tutorial. It's really inspiring. I have an application question specifically about the layers. So when working, for example, in layer fMRI, we usually just use free surfer to extract gray and white matter surface, and then we do intermediate surfaces with either equidistant or equivolume. And these not really match the histological layers because we also don't have the resolution for it, but we try to distinguish between different levels of cortical depth. Do you see any way of how you would use SIIBRA and the BigBrain Atlas to actually create layer masks for fMRI that resemble the specific region of interest, layer profile, or the histological layers more closely?
There are different ways that you can, different approaches that you can take. For some of them, I'm not an expert, but we have some experts in the room. But what you can, so first of all, since we are here at the BigBrain Workshop, we didn't spend a lot of time on this, but we have the cortical layer maps in the BigBrain, yeah? Not sure if you've seen that. So if you go here in, and these are published by Konrad Wachsstühl, so you also find volumes for download on the servers hosted at McGill, and here in SIIBRA Explorer you also can see them, yeah, just by selecting here the cortical layer parcellation in the BigBrain space. So of course you can use these cortical layer maps to measure kind of layer thickness profiles at different locations. But these are, of course, specific to the BigBrain and its folding pattern. Yeah? So it's, if you then translate them via coordinates to your standard space, they will, of course, not represent the average of the population necessarily or anything like this. So the layer thicknesses are always very dependent on the individual folding. So that's why I would not necessarily recommend to do that for your purpose. Maybe also a question to the crowd here. I mean, there are cortical layer. Are there any average cortical layer maps out there actually in standard space? Is anyone aware of that?
So we've been working a little bit on that from MRI data, but I don't think there are any average maps. But if you wanted to use these ones with your equivolume model, for example, I guess you could try to take your specific cortical parcel and map the equivolume fraction for the specific sort of layer or depth that you're looking for from these surfaces, for example. I don't know if it will. I mean, you're using the curvature and thickness of this BigBrain subject, and if the equivolume model is a good model for capturing layerification based on the volume fraction, then in principle it should work on your MRI data as well in that particular cortical region. I was actually interested in when you're saying that you have these 1 micron layer segmentations. I'm wondering if you would sort of could refit the cortical surfaces from the 20 micron ones based on sort of looking into these geometrical models for cortical aerification and sort of propagate those close to the sections where you have the 1 micron layers.
This is definitely something we will do. Yeah. So we are, of course, in touch with Konrad Wachsthühl, and this is something, the more reference points we have in BigBrain, the closer we get to do a substantial new fitting of these maps, right? So this will certainly happen on our side, and also with the data set that we are preparing, this is such a good distribution across the surface already that you could even interpolate them, and for the level of free surface surfaces, an interpolation between those will already serve. Yeah, but this, we don't have that yet. But this is something we will provide soon. Yeah.
Excellent. So surfaces were created in a way you just described.
Yeah.
So we have
Exactly.
drawn the different laminas at 1 micrometer, and then they were downscaled and calculated.
Okay. Yeah, okay.
I think I also have that here on the slides if you want to. So
We have one publication with trying to separate the deep and superficial layers from ex vivo MRI at 120 micron, which has 15 or 17 subjects, so that has some statistics about what the depth for that specific border is. If that matches your specific border of interest, that's another question. But there is something at least in that.
the data we talk about, it looks kind of like this. Yeah, you have the current data set that I already shown under the hood. You have here already now far more than 100 at least of these patches where you have the cells and you have detailed annotations for this patch. Yeah, and there's
Published 120 and new coming 860. So we have close to 1,000.
Yeah, so 120 is published, but the next version of the dataset will have approximately 1,000, which are distributed across the cortex.
And I think for the surfaces from the Wachsthühl study, we have annotated 10 areas manually. So we are quite sure that this should improve the quality of the surfaces dramatically.
Yeah. Of course, it gets much more interesting even once you go into multiple subjects, right? So in the future, we want to use the workflow that we present here also to apply to data from different subjects. But it's not as easy, you know. For the big brain, it's really, since for the big brain we have a whole brain reconstructed with all the links for the histology, we know the sections and so on. So there we can operate, and of course it's it's a lot of work to prepare more brain samples in the same or even improved fashion, but this is something that the HIBALL and BigBrain project, of course, are working on. Okay, so one thing that I briefly touched, but I didn't really explain in quite some detail is, when you work with such multi-scale data, then you always have this problem. You want to use a certain workflow, but the type of data is very different. So it's usually when you have a workflow, it's a huge difference whether you work with a 1 millimeter MRI, which you can download as a single file and have on your computer, and you can use software and just load it and look at it, as compared to something like the BigBrain, which is also a 3D image volume, but you cannot do any of this. Yeah? You cannot download the whole 3D volume. We cannot provide a Nifty at the full resolution for you to download. And even if we would do that, you would hardly find a desktop software that would allow you to load it. Yeah? That would simply not work because you don't have so much working memory, and so it doesn't work. But if you want to interact with data at multiple scales, you somehow are confronted with this problem. How do I still bring this into a similar workflow? And the approach that we have taken here, which you have briefly seen, at least in between the lines, is that we say, in the end, when you apply a certain method, you will anyway only look at a certain data size. So what we assume is there is an image resource, and there is a way to get the actual voxel data from the image resource. But if it is a very large one, then you are explicitly informed that you either operate on a lower scale version or that you have to zoom in somewhere. You have to iterate in chunks over it or whatever. And we have built this, basically this pattern here into SIIBRA. And the way that this appears is that whenever you fetch something, there's always a command fetch. You basically retrieve image data from an online resource, but sometimes you want to explicitly provide a resolution. Yeah, or a volume of interest. We have seen that in between. So what we do here, for example, is we fetch the BigBrain at a resolution of 1 millimeter. So we get a nifty file that we can, we get a nifty file that we can then actually just plot like a conventional nifty because it has a 1 millimeter resolution. And that is, of course, sufficient to look at the anatomy of the big brain from a whole brain perspective. And whenever you want to see details, just as you would do it in the viewer, if you want to look at the details, your monitor would also not allow you to see individual cells while you look at the whole brain volumes. You would have to zoom in, and when you zoom in, you only see a part of the brain anymore. Yeah? While you sometimes keep in a second window the 3D overview. And it is more or less the same idea here. So if you want to operate on a higher resolution, then you usually define a volume of interest, or what you might call it, a bounding box. Here in this example in the documentation, we just provide 2 points that span the bounding box. It's an axis-aligned bounding box, so you just need 2 points here. So we define a bounding box in the BigBrain space, and then we communicate with the same object, the BigBrain template. Here, it was also the BigBrain template. Here we fetched the whole volume at 1 millimeter resolution from the BigBrain template. Here we fetch only this volume of interest on this little bounding box, but now we fetch at a resolution of 20 micron. Yeah? So we are talking to the same object. The object represents the resource, the BigBrain, as it is stored somewhere on a server in the cloud for you, and we just communicate with it. Yeah? Now we want something here with a high resolution, now we want the whole thing at the lower resolution, and this all produces nifty images that I can superimpose because they live in the same space. Yeah? So here we look at this small chunk at a full resolution. The contrast is not so nice here in the viewing, but this is now full BigBrain resolution. And we can also plot both of them. So I can plot the chunk and I can plot the whole volume on top of each other. I'm coloring the chunk now with a different color map so that I see it. And now you see that the little chunk is sitting somewhere. You almost cannot see it. I make this a bit. You see this little chunk here. Yeah, that's the one that we were looking at here. This little one, but if I plot it together with the 1 millimeter version of the whole BigBrain, and I plot it in here in this magma color map, you see that it's just this little part here, but it's, you know, just by having the nifty images with the spatial information, they still belong to each other, yeah? But it's different resolutions. I mean, usually when you work with neuroimaging data, you know these concepts, but often the differences in scale are not that dramatic, yeah? But of course, you know that you can also have a 0.5 millimeter MRI and 1 millimeter MRI, and you use the same concepts, of course. So actually, it becomes more important to keep track of the protocol, how you receive the data, than to actually store the actual nifty file. Yeah, that's kind of an important message. So when we work with this high resolution data, it is always important to remember how to get it than to actually have it, since you can anyway not always keep all of the image data for yourself on all your machines and so on. You have to somehow rely that it's somewhere in the cloud, and you need to find ways to remember how to get it. And that is what SIIBRA solves here at this stage. And then same thing. We have now used the same image resource. We used different protocols to get image data, high resolution, low resolution, but it was the same image resource. And now the second case is once we know the protocol, how we actually got the high resolution small part of the big brain, we can also use this protocol and apply it to a different resource. For example, the cortical layer maps. These cortical layer maps are also very huge, but it's a different resource. It's a different image data set somewhere out there in the cloud. Yeah? But I can now use the same protocol, namely this volume of interest and the specification of the resolution, to now also get data from this different resource. That's what we do here. Yeah? We use a different resource, the cortical layer maps, and we fetch using a protocol and reusing the volume of interest definition that we also used above. So here we defined this volume of interest. We used it to fetch from the big brain itself. And now we use the same volume of interest definition, and we use it to fetch from a different resource from the cortical layer maps. What we get are two nifty files with a zoomed-in location in the big brain. One has the cortical layer mask, one has the big brain data. And again, they fit to each other. I can use them together. So here we are plotting them on top of each other. This is the color map, don't get confused, but you see that actually the retrieved cortical layer map and the full resolution small big brain chunk, they fit to each other. Yeah? So I have a set of two nifties with the cortical layer maps, and it's not the full big brain, but it's the full resolution. Other things. So one thing that we did not go into much detail is how you find regional data features that are attached to brain regions. And actually that is a large part. So in eBrains, where we curate a lot of data, where we integrate a lot of data from different labs, for the largest part, this data is linked to certain brain regions. So very often people come with a study, with a collection of data, let's say from a histological experiment, and they say, okay, this is from the hippocampus. Is a piece of tissue that's from the hippocampus, and we do not have a way of registering the image, yeah? Or it's even just some average densities measured somewhere, and then usually we just know the name of a brain region. And so searching by brain regions is something that gives you much, much more data than just exploring the space and look for registered image data. And something that is used a lot are these neurotransmitter receptor densities. So the way that you can search for these, they have their own modality, of course, molecular receptor density fingerprints in that case. And we can here use a parcellation again. We use the Jülich brain parcellation to basically ask, are there any receptor densities measured for any brain areas defined in the Jülich brain parcellation. So this is what I called in my slides a semantic query. And then I get a list of fingerprints. Sorry. And for any of those fingerprints, here it's the first one, I can look at the data attribute, and you see that this data attribute is a data frame. It's a kind of an Excel table. It's a pandas data frame for those of you who work more with Python, with mean and standard deviations of different receptors. And often when you find such regional measurements, very often in your workflow, you actually want to have them back in the 3D space. So you want to see them on the brain or something. So what we do quite often, what users do quite often is actually then create a map of these densities. And this is also easily done. So what you can do in SIIBRA is, for example, you usually compute a dictionary structure which basically maps a brain region name or a region object to a certain value, which is here the average receptor density. So what we did here is we went through all the available receptor density fingerprints, so these tables here. We looked at one particular receptor, GABA A, and we collected these average values for all the available brain regions in the Jülich brain. Yeah? So we built a dictionary that says Jülich brain region was this and that average receptor density. And then from this, we can then colorize the map. So what we do here is we retrieve the M&I map of Jülich brain, and then we colorize it using that mapping, using this dictionary that tells which density for which brain area. Then we have a brain volume, And as I always say, this brain volume is not yet data. It only describes how to produce or get data. And once we say fetch, what it actually does is then it downloads the Jülich brain map and it modifies it with the new colors. And then I can look at that map. That's what you see here. And what you also see is this is incomplete now, because we have only colors for some regions. We have only measurements of receptor densities for some of the regions, not for all of the regions in the atlas. That's why we get now here a map object which has values for some brain regions and gaps for others, of course. And you might decide for yourself, if you use that, whether you interpret the gaps as gaps or whether you want to interpolate between them. I have seen people doing both. I would, of course, advise to be very careful interpolating such large gaps here. In the consortium here, we are also working on whole brain distributions from autoradiography. There will, I hope also rather in the nearer future, there will also be whole brain volumes with interpolated cell densities from autoradiography, where you can really then sample receptor densities at arbitrary locations. There's a publication out on by Nicola Palomero Galafa and Thomas Funk, who are both part here of the BigBrain project already, on how these are computed, and these volumes will at some stage also appear here, so you would not have to go over brain region averages to see receptor densities on the brain surface or on the brain volume. Here. Okay, maybe maximum one last example. If you're interested in connectivity matrices, things are just the same. So when the most frequent instance of connectivity matrices that we have linked with SIIBRA are parcellation averaged ones. And then, of course, if you want to search for such matrices, connectivity matrices, you would search by parcellation. And that works exactly the same way. Yeah. So you would again ask to get features. There is something that I click on here accidentally all the time. And you provide a parcellation. Here it's again the Jülich brain. You provide as a modality now one type of connectivity, streamline counts in that case. And then you get a set of features where streamline count data has been reduced to the Jülich brain maps for different cohorts. Here we select one from the Human Connectome project. You see there's 200 different matrices here based on that. And when you fetch from these features, you also get a data frame with a connectivity matrix, which has the region objects as column and row headers. And you can then, of course, look at these matrices, plot them on the brain surface and so on. And there's quite something available here. So from different cohorts, 1,000 brains, HCP, and so on, and different subjects, streamline counts, streamline lengths, there's functional connectivity. So there's quite some connectivity information here, and it's, of course, also growing. We benefit a lot here from the standardization efforts that are currently going on regarding bits and so on. So I actually hope that we can more share such data sets between the different repositories. We are using eBrains here a lot, but the more we standardize these data sets, we can, of course, make them accessible in the different systems. All right, maybe then I think we are approaching the end of the extended time.
Can I ask one question quickly?
One more question, yeah.
Can you have access to the transformations that you sort between the spaces?
Yes. So the transformations that we used, they have been computed in France, and actually one of the prominent persons that were doing it is here in the building, Jan Le Prince. He's sitting over there. Yeah. So they are actually that's a nonlinear diffeomorphisms that have been computed, but they have been they have been guided by sulcal patterns. So they are somewhat constrained by the sulcal patterns of BigBrain, M&I space, and so on. And there's also actually a website for this. And Jan, you can correct me if there would be better resources to go there. It's not very often asked, but here's a GitHub page which describes actually the code that we are using as a backend service to do these transformations. And there you can also download a Docker image. Is this the right one? I think it is, yes. It is, yeah. So there's also on this GitHub page, there's also a description what the methods were to do this with the publications. And there's also a Docker image which you can use to warp images yourself and so on, yeah. We are using an API that can actually, we send coordinates and it sends back coordinates in different spaces. We're using this a lot in SIIBRA Explorer, but you can also run this for yourself with a Docker. And hopefully give credit. I think that was Jessica Levenberg in that case, one of the main authors of these transformations between BigBrain and the other templates. Yeah, so that's available. It's a diffeomorphism. Yeah. You will sometimes when you use this. I didn't show that in SIIBRA. Actually, I didn't show that at all, which is a pity. So the way that this is modeled in Python. That's very fast. Let me just, but it's nice. So if I have a point I can see SIIBRA point, and then I can say 1, 2, 3, space is M&I. By the way, these spaces, if you wonder how I come up with the names, I can always go the proper way. So there is a registry, SIIBRA.spaces, and then I can tab complete, and then you can see that here is M&I. So when I write M&I 152, it's just a short form of finding this this particular thing here. And I say here is point in M&I space. So now I have created this point. Yeah, you see, it has stored this. And what I can then do is I can say warp, and I warp this to SIIBRA.spaces. BigBrain. And now I get a different point in a different space. Yeah? And this is actually what it's actually doing, it's calling that backend. Yeah? And I can do this with bounding boxes and all sorts of stuff. Usually not directly with images because that's an involved computation. So it's SIIBRA doesn't do on-the-fly warping of images, which is possible, but it usually warps coordinates, bounding boxes, things like this. Yeah.
What about grid? Can you just give it a grid?
Sorry?
Can you give it a grid?
What?
A grid of points. Like an image grid.
A grid, yeah. So, sorry, I didn't get it. So what we use here, SIIBRA.point, this is one form of location in SIIBRA. And you see here, if I go to locations, there are bounding boxes, there are contours, which are actually lists of points, there are point clouds, sets of points, so there are several objects. There are also bounding boxes we saw already, but in the end it's always a set of coordinates which can have an uncertainty, and they are always explicitly linked to a space or explicitly linked to no space. So you can also say, but if you want to create an object, even a point, and you don't specify anything, it will complain. Yeah. Oh, it should. No, but it says space is none. Yeah, but actually, in some cases, it will even complain that you should say something. Yeah, but it's usually SIIBRA keeps track that if something is in a reference space, that this information is also preserved. Yeah, and the objects are all based on points with different constraints. A point cloud is an unstructured set of points. Bounding box is, of course, a set of corner points, and so on and so forth. Yeah. But this should work. If it's really huge, it takes a bit because this is really done in live interaction with the backend. And what you can also encounter is an interesting discussion that Jan and I also had recently. I think actually there's an update in the making. Sometimes specifically if you have bounding boxes, or if you consider this 1 micron, these brain sections that we have, yeah, such an image, the corner points are actually far outside the brain. So if you have such an image section, a coronal section, which usually looks like this, and then you want to warp the bounding box. It's actually tricky because the points are outside of the brain, and the transformation usually don't know what to do with points outside the brain. Yeah. So you could run into such issues that it tells you this point could not be warped because it's not out inside the brain. There's actually we're actually working. I think there was an update recently, even that we try to regularize even outside the brain, so that you do reasonable things, although it's not clearly defined, even if you're a little bit outside. All right. If you don't have more questions, I think then we stop here, having filled the 2 hours, and I thank you very much for your attention. I think everybody was quite attentive, at least it appeared to me that you were attentive, you were still looking at me. So thanks a lot for your attention, I hope you enjoyed it, and if you have questions, just come back to us. Everything that you saw is always in development, and there are updates coming and more data is coming and things might not work. So if you like it and if you use it and if something doesn't work, please let us know. We are happy to help, and we know that not always everything works. Thanks a lot.
Susanne just said that there will be a coffee break for about 1 hour because we have some 6 speakers. So therefore 1 hour coffee break, and then we will continue with the normal schedule. Okay?
Thanks.